Our next speaker is Nikolai
Prokhorov.
Say it.
Prokhorov.
That. He is going to tell us a little bit about Lambda phage structure and host recognition. I'm sorry for butchering your name.
That's fine. Okay. Should I do something for it to appear? Presentation. Okay, here it is. Good afternoon. Thank you for having me here. And I would like to share some of our work on Lambda phage structure and host recognition. So the first question, why Lambda? And we found two reasons sufficient. The first, Lambda is a fairly well-known phage. So over the 70 years or so, quite a bit of work been done, but it's kind of clearly understudied structurally. So that's a nice target. And the other reason is that it is well known that this phage, in contrast to so many others, can kind of skip all the auxiliary interactions with the cell and goes strictly to the ultimate receptor, which is outer membrane protein Lambda, and that's its interaction with Lambda is sufficient for the particle opening and the genome transfer start. So that was the second reason. We started with a fairly clean phage, in contrast to so many in academia, I guess, and we collected quite a bit of data, and since the tail is flexible, so we used the CryoSpark filament tracing pipeline to pick the particles, and we've got some really good tube classes, like 1 million of particles or so, and we've got some kind of not so good tube classes with recognizable features, nevertheless, and from those we've got particles for the neck of the intact phage, for the neck of the empty phage, which spontaneously released its DNA, and particles for tail tip assembly. So first we reconstructed the tube, and in C6, the tube went to pretty high resolution, but it recapitulated the structure, the intermediate resolution structure, which was established before, more or less, but at the low contour level, we would see the periphery of the tube. And it's important to know that the tube protein GPV consists of two domains, the N-terminal domain, which is the tube itself, and the C-terminal, which is IG-like domain, basically beta sandwich. And we realized that something is off because we have 6 tube domains and basically 12 IG-like domains. So we've done C3 reconstruction, and the periphery now is much better. So we have 6, the right number of IG-like domains. The important part here is that we can see at very low resolution what we identify as a tape measure protein, and here it's much more consistent with the kind of hexameric state, in contrast to what's been known so far for some Sypha viruses. Yeah, and it seems the same way both in C6 and the C3 reconstructions. Okay, so we've built a model for both domains, and it looks like that IgI-like domains, they like folded hands. They prefer to be in pairs, and if one pair is formed, the other two are kind of forced to be formed too. And but it happens independently in every ring of the tail. That's why 2D classes look a little bit messy, because it's kind of stochastic and chaotic. But yeah, after that we reconstructed the neck assembly, and here is you can see the C6 map. In the asymmetric unit, the full assembly consists of 5 or 4 protein, depending on whether you want to count to consider the tube protein part of the assembly. In the full assembly, it's like 12 portal protein copies, 12 head completion protein copies, protein W copies, 6 F2 junction protein copies, and 6 tail completion protein U. So the most interesting part is it? Why did it stop? The most interesting part is the interface in between the W protein and F2 protein. F2 has like pretty it has tube-like fold with one addition of the alpha helix, and 6 alpha helices form a kind of perfect collar, a ring. And the tail completion protein forms 24-stranded beta barrel, which fits into the collar nicely, and that's how the interaction is organized. So the interesting part about the NAC assembly is that the DNA seems to be kind of unengaged and not interact very much with the tube itself. So the narrowest point is this beta barrel of the head completion protein W, but the density for the DNA does not seem to be affected. And we believe that the NAC is actually, and this is consistent with the phage biology because it looks like it tends to eject its DNA as quickly as possible to circularize it and prepare for integration. And that's why the NAC assembly evolved the way, so not to impede the ejection process. That's our understanding. We reconstructed the empty NAC, and it's pretty much the same, again, consistent with the idea that NAC is not very much involved in the ejection process, and yeah, with the only difference is the absence of DNA in it, obviously. So after that, we reconstructed the tail tip assembly, and it consists of 7 or, again, 6 proteins. Depending on whether you include tube or not. The tube is hexameric. It sits on top of the hexameric assembly of the M tube-initiated protein, and that's how it's organized pretty much in all studied Schitoviridae to date. But the rest of the particles is pretty much kind of Lambda specific. So we see here the next ring is pseudohexameric, and it's composed of 2 proteins. The tube-like domain of the giant straight fiber protein, and the protein L, which is a homolog of hub protein. But here it's not perfectly a hub, right? So we call it quasi-hub protein. So they form this pseudohexameric ring, which accommodates the symmetry mismatch between the hexameric part upwards, whatever upstream, and the perfectly trimeric downstream. So the L protein donates a little kind of domain into the next, which is not a ring anymore. It's a threefold structure composed of the unrelated domains of the straight fiber. And the straight fiber is much longer than what we see here. The C-terminal part is quite nicely predicted by the AlphaFold. It has like the beta helix, and it's supposed to end up with a bunch of IG-like domains. So we identified the minor protein I, and we call it ripcord, in the tail. And the other thing, which is kind of important, we see the very C-terminal alpha helix of the tape measure protein. And in this case, it's like in other phages, it's trimeric, which is not exactly consistent with what we see in the tube. It's trimeric in form like this tri-helical bundle right in the middle. So we find like 3 proteins of this assembly especially interesting. And oh yeah, I completely forgot about that. I mean, it's been known that Lambda has 4 lateral fibers, and some people kind of consider that it's probably some kind of an artifact, and a couple of fibers were lost during purification or whatever. But our structure actually shows how the 4 fibers can be incorporated in the overall 6-fold structure, because we see the collar around the tube-initiated protein, which consists of 12 domains, and those are exactly the N-terminal domains of the STF protein, which forms the proximal part of the long fiber. So 3 of them can form the trimer, and that gives us 4 fibers per particle. Okay, and that's the. Why didn't it work? Yeah. Okay. So okay, that's the most interesting part of the structure from our viewpoint. So here you see the giant protein J, the fiber, and the trihelical bundle of the tape measure protein, and you can see that the protein I actually tightly interacts with both of them. So it's and at the same time it forms like a core, which is at the same time forms the bottom of the lumen of the tube. So and the C-terminal part of the protein I goes out of the particle and donates a beta strand to this really peculiar immunoglobulin-like domain of the GPJ protein, and it's interesting because it's covalently linked on the top, and all the rest is just kind of stick to the particle. And we expect that it can kind of be quite flexible, and for example, the interaction with the receptor could kind of pull it away, producing enough pulling forces to kind of unfold the core of the ripcord protein. So that's how, and eventually to kind of open the particle. That's our hypothesis. And to we decided to kind of to get the post-attachment structure, and for that we need the receptor and the phage. So we cloned the receptor, we expressed it in inclusion bodies, we kind of refolded it, and in a couple of steps of purifications with the affinity purification and size exclusion purification, which actually both of them shows that there is quite a bit of heterogeneity in the sample, because not all of the protein even bound to the resin, and the size exclusion profile, though reproducible, is quite characteristic with some weird peaks, and the luckily quite easily recognizable kind of monomer peak by its size. So we tested these fractions, and weird fractions did not affect the phage at all, but the monomer is actually inactivated that, so we decided that the folding worked well, refolding worked well, and that was nice because we basically kind of tried the first refolding protocols for outer membrane proteins we found, we Googled, and that reminded me of the famous thought of the Leo Tolstoy about the happy families being all alike and unhappy families being unhappy in their own ways. So it looks like these outer membrane porins, proteins, they are more like happy families, have much in common. So, and after that, just to avoid the horrible mess of visualization of phage pumping out DNA, we made a tail preparations. For that, we killed the capsid gene. We sampled tails and a kind of few steps of fairly straightforward purification, like differential centrifugation, anion exchange chromatography, and like sucrose gradient. We've got a pretty decent sample of the tails, just slightly contaminated with the GROL. And as we can see here on the negative stain, and we mixed them and collected some data, and the 2D classes looked kind of pretty promising. So here you see that they are kind of mostly empty. They are lacking their tape measure protein. We can see that they are kind of open, and we see a completely new structure appearing on the side, which we recognize as the tail tip, tip of the straight fiber coming all the way upwards. So, and the reconstruction seemed pretty promising. The curves looked good, but the structure didn't. So apparently the microscope didn't work well that particular day. So whatever, and the structure cannot be so it's like nominal resolution is high, but we do not have features. But we kind of still can interpret it in terms of domains, and again we see that the J protein kind of opens up. The channel is empty. We see some detergents here, and the tip of the straight fiber appears. That looks like a predicted beta helix, and even some immunoglobulin-like domains from the tail tip are recognizable in here. So we're obviously working on getting kind of decent data and get high-resolution structure. But the interesting thing is that if we compare pre-attachment and post-attachment structures, they kind of seem fairly similar in here, except for this peculiar immunoglobulin domain connected to the ripcord, which is in the post-attachment structure is the density is absent, which is probably, it's unlikely that it's unfolded. Most likely it's really flexible. That kind of points on the possibility of its engagement into the interaction with the receptor. So, and in the world of Lambda, nothing is completely new. So some approaches to a kind of visualization of that type of thing have been made. So in Texas A&M in early 80s, people studied that. So they used two versions of the Lambda protein. They reconstituted it in liposomes, and the difference between these two versions of the Lambda is that this one does not inactivate Phage unless chloroform is added, and this one inactivates Phage. In our research, we used this version, and we've got pretty much something like here. But when people in Texas A&M used the other version of the receptor, they kind of imaged that only the very tail tip, the tip of the fiber, interacts with the vesicle. So we think that's like a good indication that the interaction of the Lambda with its receptor is kind of a little bit more complex, and it probably goes in two steps. And we're going to kind of investigate it further. So this is a side kind of project of our standing DNA packaging collaboration between Mark Moret's group in Indiana University and Carlos Catalana in University of Colorado. Thank you.
We have a couple minutes for questions.
Thank you. Lovely to see that things can be kind of translated to. You don't reconstitute your Lambda in any nanodisk. It's just in detergent.
So I guess that's warranted now, right? We will try and do it, but yeah, here it's just detergent. And I think that for kind of these intermediate not stabilized by the interaction with the membrane, it seems kind of pretty solid. So I'm quite hopeful that our nanodisk would help kind of a lot.
Well done.
Great structure, Nikolai. I'm curious about the DNA in the neck region, where there is so much space apparently between what you identified as DNA density and the outer wall or the inner wall of the neck. If the DNA is actually pushing down from top, from the bolus of DNA in the head, you would expect that thing to sort of deviate from just this straight line without anything actually holding it in the central position. Or it's actually being restrained at the top, and that's just some dangling end that has no attraction or repulsion at all, and of course then stays centered. Do you have an opinion on one way or the other where the DNA is actually anchored in the neck to leave it in that position where it has no contact with the inner wall?
Yeah, it doesn't look like the DNA is anchored in the neck, I mean, from my opinion, because we have some solid structures of the phage when the DNA is kind of in the neck and it's fairly distorted. It usually goes kind of like that. It interacts with some charged residues like arginines or whatever. It's interesting that it's a narrow part of the barrel. There is actually an arginine kind, and it's quite a bit of them, but they are. But the DNA density is not distorted, and right next to this arginine there is like glutamate, so which probably kind of compensate its positive charge. And I mean, it's well known that Lambda DNA goes very deep into the tail, right? And that's what we see in our reconstructions. It probably goes like 30% of the tail length.
The DNA in the neck has been incoherently averaged as well. I mean, we ever did an asymmetric construction, I'd see contact. Yeah,
I mean, maybe, but I find it unlikely.
Other questions? All right, let's thank Nikolai again.
Thank you.
Our next speaker is Pavol Bardy, and he's going to be telling us about a single-stranded DNA virus genome delivery.
Thank you, Carol, and thank you organizers for inviting or giving me the opportunity to present the research in form of a talk. And so far we see quite a lot of things about phages which have tails, but I will be presenting something different. And it's about unrevealing a mechanism how genome is delivered from ssDNA model virus, which is actually a phage belonging to Microviridae family. And single-stranded DNA viruses are, of course, important. They infect humans, they infect animals, and they infect plants, so causing huge economic losses each year. And then also we have some quite useful models, as is Phage 174, and adeno-associated virus, which is used for gene and cargo delivery. And the thing is that we actually don't know that much about mechanistic of these viruses and their variants, how they function, how they deliver genome. The only well-described case is the Phage 174. And what we have here is actually icosahedron in T=1 architecture, which is decorated on 5-fold vertices with spike receptor binding proteins. They recognize the host. After host recognition, internal features in the virion got reorganized, and ejection of H protein, which creates some kind of decameric tunnel through the cell envelope that is ejected, and with that, through the tunnel, also the genome. Now, while this is a good model, it covers only a very specific niche of microviruses, mostly polyphages. We don't know much about the other ones. So here I'm presenting a very simplified taxonomy of microviruses. So these are similar to Phage 174. And these differs because they don't have receptor binding protein assembled on the vertex of the capsid, on the 5-fold vertex of the capsid. Rather, they have extended the loop of the major capsid protein, and it creates protrusions on the trimeric sides of the virion. And these viruses, we don't have that many models. This structure, which I'm showing here in the green, that's for spiroplasma virus, but that was only built in C alpha because the resolution wasn't high enough. And very recently, Bentley Fane with some collaborators, they solved one coliphage structure. However, there is much more to discover, and it was last year also shown that actually microviruses are very important inhabitants of human gut, similarly as Oli was showing his research on the KRAS phage, and he told us that healthy human gut are actually somehow interlinked with occurrence of a KRAS phage. In case of microviruses, it was shown that if there is an occurrence of microviruses, the cognitive function of the individual are actually declining. So you don't want microvirus in your gut. And here, a new model system, which I'm about to announce and to present to you, Rhodobacterphage VM1. Nobody knows how it looks like because I isolated it and nobody else saw it. So actually, it's a microvirus with the longest genome discovered yet. So it has more than 6.6 kilobases. And it's indeed a single-stranded DNA virus, as you can see on the gel. When you treat it with DNase and with mango bean nuclease, which cleaves any single-stranded nucleic acid, you get nothing. But if you treat it with RNase, you still have a band. And here you see the circular map of its genome. So what is actually, why it's so big? Well, it's big because it contains endolysin, and this endolysin is functional. Maybe it's not that well visible here, but believe me, it is. So far, I think it's the first microvirus that was proven to actually have functional endolysin encoding. So this was done in zymogram on purified cell walls of Rhodobacter. Also, it encodes DNA methylase, and it has 5 methyl cytosine in the genome, as was verified by nanopore sequencing. So today you could already hear something about Oxford nanopore sequencing. And actually, if you want to sequence single-stranded DNA viruses, I can highly recommend. It's a very accurate method, and it can provide you with quite useful information. So this is how the actual structure looks like. So you have T equals 1 capsid, as I already told you. That's a common feature in single-stranded DNA viruses. This is a native particle. Around 3 angstrom resolution, and those protrusions, which you can see, they are actually on the threefold axis of symmetry, and they are composed out of the major capsid protein spikes. So this is how the capsid protein looks like. It has this classical fold of single-stranded DNA viruses, and the loop, which is positioned at the, all right, here. That's the 50, something around 50, 60 residues couldn't be modeled, and that's the actual protrusion, which you can see in the map. It just doesn't have, of course, because it's flexible, it doesn't have that good resolution. So when we compare it to the other structures of microviruses that were solved, there are some striking differences. This is the structure solved by Bentley Fane and collaborators. And what they saw in their structure was that there is this N-terminal extension which somehow stabilized the pentameric capsomer formation. Now, in FIM1, this is not occurring. However, there is C-terminal extension which basically serves the same function. It binds to different parts of the major capsid protein, but more or less the function is the same, and that is to stabilize the virion. In case of FIX174 and other viruses, none of these extensions are present. So that's quite interesting observation. And here I'm just wanted to show that RMSD between FIM1 and this model, that they are basically the same fold. It's very, very similar. So apart from this extension, nothing is that interesting. This is an alpha-fold model of the actual spike. So as I say, you can see the density here gets very unspecific, but one of the models predicted by AlphaFold actually fitted quite well. And here you can see when you morph the best rank models, you could actually see what kind of flexibility is occurring in that spike, which might be the cause of why you can't actually get it to high resolution, and also probably functionally very important because the virus wants to reach its receptor, so it has to have some kind of flexibility in the receptor binding protein. So another thing, so the native particle, that was purified by cesium chloride centrifugation. Now, I wanted to see some mechanistic of this virus, so I managed to purify an empty virion, and this was done by sucrose centrifugation. How is this possible? I don't know, but during blotting, when the virus is purified with the sucrose, it just releases DNA and it's empty. So again, 3.3 angstrom structure, and well, there is massive difference, of course, and that is there are no spikes. So the density is just gone. Even if you increase the threshold of the in chimera, it's gone. It's just completely unfolds and flies somewhere in the space. This is actually the RMSD between C-alpha atoms compared between full and or native and empty particle. So the blue means there is basically no difference, and the only difference is in the spike region where it gets, well, because the density, of course, it's worse, and then it's just not visible anymore. So after that, I isolated the mutant, which formed turbid plaques, and after doing cesium chloride centrifugation and purification, when I put this sample on grid, what happened was that I actually got a sample which was releasing the genome directly on the grids. And when I did this was actually done by my student Connor. When Connor done QD classes, he discovered that actually some 2D classes were missing part of the density. And reconstructing this in C5 showed that one pentamer is missing in around 10% of the data set. If you think this sounds like something done by Plevka lab, you're right. So they observed something very similar in single-stranded RNA viruses and did some simulations which showing that actually this kind of pentameric expel might be important for genome ejection. However, to my knowledge, there is no evidence this is biologically relevant for the actual genome delivery. When you have virus and you infect host, it's an actual thing that it's happening. It's not just artifact of in vitro disassembly of the virus. So what I wanted to do was to do tomography and see how it looks like when this single-stranded microvirus is infecting cells. So most of the people would tell you, you can't do tomography on cells and not mill, and for sure you can't do it on 200 kV microscope without an energy filter. And because you get something like this, which is just very rough shape of a bacteria cell and a lot of noise if you use dose, which is usually used for single tilt for tilt series. However, I decided I won't go for a very time-consuming milling, and I will use my 200 kV microscope and rather optimize cultivating conditions to obtain thin cells. So what I do, switch from rich media to minimal media, then edit a molecule which is a quorum sensing signal of Rhodobacter, and it's actually causing the cells to grow smaller. And then also we applied some kind of centrifugation to get rid of the very large cells and enrich the very small ones. So now, with the same dose, you can actually see features quite well. Another issue was that the distribution of the cells, so this is electron microscopic grid, and this is individual grid squares, and these black objects, those are Rhodobacter cells. And as you can see, not that many of them. So luckily, I was trained by the Eva, who will give a talk right after me, and she was very competent and gave me very good advices, and this resulted in much more abundance of cells on the grid. So after this, I feel confident enough to mix my virus with cells and collect some tomograms. So here you can see one of them. Here is a cell, and here are viruses attached on the cell. Some of them are empty, so around 10% are attached are empty. And if you paid attention, you know the empty ones no longer contain spikes, so it means they're actually in the process of genome delivery. Otherwise, they wouldn't bind to the bacteria. And indeed, if you then look around, I don't know, carbon areas and places where I did tracking and focusing, you'll find out there is plenty of empty particles which are not attached, while all the full one, they are attached on cells. So I collected several tomograms, and good thing is that in every case, you could see viruses attached right directly on the membrane. This is a bit striking because Rhodobacter actually contains capsule. And for example, in the case of Rhodobacter gene transfer agent, they can never reach a membrane. They always keep to be stuck on capsule. However, this microvirus has absolutely no problem with it. It doesn't contain any enzyme. It has just major capsid protein with receptor binding loop. And for somehow, it can breach this protection. So anybody of you have any idea? I'm glad to hear how it's doing this. So I collected more than 50 tomograms. From these, I got more than 800 particles. And then I switched to subtomogram averaging. And I would like to stall you and present you amazing results which I got, but so far I don't believe they are true. It looks like overfit, oversharpened initial model. So I'm doing something wrong. So the conclusion of my talks, VM1 is a new model of microvirus with the largest genome yet. And it has a unique C-terminal extension of its major capsid protein, which is somehow stabilizing the whole virion. Then there are trimeric spikes, which recognize the host. So actually, also different mutants I obtained, I sequenced them with, again, nanopore, and I discovered how they differ, and they actually differ in the sequence, in the spike. So there's like additional evidence that this is indeed the part which is important for host range. And yes, they disassemble when the genome is released, and based on the tomograms, I also know once you have empty particle, they are not longer binding to cells. Pentamer expel is observed in vitro. Now, questionable if this is biologically relevant. And somehow the virus is able to breach the protection by capsule of its host. And outlook, what needs to be done is to perform the subtomogram averaging of the data I have, and hopefully that will close the story. So I would like to thank the whole YSBL, the organizers, for putting all this event together, because I know I only co-organized online one, and I know it's quite difficult. So I'm really glad they did this task. Crown facility in New York, Jamie, who is the head of the facility and is trying to push the tomography there quite a lot. Then so of course, Fred, who is my line manager in chemistry, Paul, who is my line manager in biology department, and collaborator is doing the nanopore sequencing, and then Wachu, whose lab I could visit to learn how to process tomograms, and also Alan, who is actually my mentor, whom I met in PVA 2019. So Fred I met in PVA 2019 as well. So all the people I know and who I'm collaborating, I met on PVA. So great conference. Thank you.
Questions?
Great talk, Pavol. I had a question about, it's a comment about the fact that you can see your viruses attached to the cell. Do you think it's probably because they are stressed because you grow them in minimal media and now they don't have that mucus layer, whatever it is?
Yes, so these are basically done in very same conditions. Okay. Then I added this acetylserine, but I repeated it, and again it looks the same. Yeah.
Maybe I'm imagining something. When I looked at your in situ tomograms, are the ejected capsids seem to be associated with the inner envelope? Are they compartmentalized, or are they random distributed throughout the cell, or am I just seeing things?
Oh, these. So they are more or less, I would say, randomly positioned, yeah. And they don't seem to be internalized because I'll show the tomogram again. Oh, is it playing? Yeah. So somewhere will be empty one. You can try to catch them here. So they seem just to release the genome, and that's it. Yeah. Yeah, one more time now. Here are some. Yeah. And the things inside, it's just some spheroplast. That's not virus. Yeah.
Yeah. So about the spike protein. So is the idea that, as you said, it once you bind the host, it disassembles and flies into space, and then in the tomogram, if I remember correctly, you said that you see that the virions that have bound have kind of lost all of their spikes. So how would that work then? I assume that you bind the receptor from one
yes.
or two receptors from one side. How do you then throw out the stuff from the other side of the virion?
Yes, so what I think is happening is that as soon as the DNA gets out, something happens which just makes this, because this trimeric structure is probably not, is like metastable, so probably it just somehow dissociates.
Over there.
So I had something similar, but I thought somehow the spikes falls off and then DNA comes out, but that's not the case.
Yeah, here they can't fall off because they basically have, it's one protein, it's like peptide bond, so yeah. If it makes sense that these spikes, they are basically, so this thing, this is continuing, so it's like one protein. Yeah, so it can't just fall off. Does it make sense? Yeah.
Hi. So my question is, you said that you were able to run gradients and you saw a virion peak and then a minor peak. Is that correct?
Sorry, can you repeat it?
You said that you ran a gradient?
Yeah.
And you saw a major peak and a minor peak? Or no?
No. So I ran sucrose gradient, and in those conditions, when I prepared a sample, it just lost all the DNA during the blotting. When I did cesium gradient, it retained the DNA during the blotting, even though it was, of course, like buffer transferred and everything. So I don't know what's going on. Yeah, but
okay. So my real question is, how can you separate like mispackaging events versus actually empty particles that you know actually like release their genome?
So in the first sample, there were like no empty particles, they were just native one. In the second sample, there were just empty one, there were no full ones. So yeah, I don't know, maybe we can discuss later, maybe I don't, yeah.
Have you just tried taking the LPS, purifying the LPS from your bug? Because in Phage, that's how we did it. It forms beautiful little membranes, and it might be a lot easier than working with the cells initially.
Might be.
Have you tried?
Haven't tried.
It's, you might have to boil it in phenol and ethane. You need a good hood. But some of these kits may work, and I would try that first before the why would it do that? E. coli is so much easier.
But
I didn't know about your capsule.
But the thing is that it's really proof of concept of thing that doing tomographic study on whole microbes, it's actually possible and useful. Yeah.
All right, let's thank Pavol again. We should move on.